patient might develop a metachronous polyp, and if that is left undetected, that could go on to progress to a cancer. Therefore, in the UK, we have bowel screening programmes, and today I'm going to talk to you a bit more about the Scottish bowel screening programme, where at the age of 50, you receive a test through the post. This is called a quantitative fit test, and you are requested to send a sample back of your stool back to a central laboratory, which in Scotland's based in Dundee, where they test for blood within your stool. If you have a positive Q-fit test, you're then invited for a screening colonoscopy. At these screening colonoscopies, if any polyps are identified, they are excised, and we use guidelines called the British Society of Gastrology 2020 guidelines to identify the patients that are at high risk of developing a metachronous polyp. So at the moment, what the BSG 2020 guidelines say is you're at high risk if you have greater than 2 pre-malignant polyps, including one that is an advanced polyp, or if you have greater than 5 pre-malignant polyps. And although these BSG guidelines give us a tool, they're at the moment not perfect. So around half of the patients are classified as being low risk. So they are discharged from the bowel screening programme and they go back into the normal screening programme, where they'll be invited to participate in a Q-fit test once every two years. However, the problem with these patients are around 60% of these patients actually do develop further polyps. Therefore, these polyps are not being picked up in the surveillance colonoscopy regime, and they may not be detected till clinical symptoms arrives, or if on their next round of Q-fit tests, they're identified as having a positive Q-fit test. So there's about 60% of patients that are being misclassified at the moment as low risk, who should really be classified as high risk. Similarly, those patients that are classified as high risk are also misclassified. So if you're high risk, you're invited to participate in surveillance colonoscopy at 3 years to check that no further polyps have developed since you were last scoped. And these high risk patients, around 50% of them don't develop a future polyp. Therefore, they're receiving these unnecessary colonoscopies. These colonoscopies are not only costly, but more important, they're invasive to the patient and they're not without side effects. So we've got about 50% of the patients receiving these surveillance colonoscopies that don't actually develop future polyps. So we're putting a lot of resource and energy into patients that should actually be classified as high risk, which ultimately takes away some of the resources that we could be putting into the true high risk patients to give much more targeted surveillance. Many patients find the colonoscopy procedure unpleasant. It is uncomfortable, but it also has 3 days where the patients could even have lost days at work. 2 days prior to the procedure, patients are asked to eat only plain foods. Then on the day prior to the procedure, they really can't go to work or leave the house because they have to drink laxatives throughout the day and need to stay close to a toilet. Then on the day of the actual procedure, they start with fasting, they might be offered sedation, and they might experience side effects. And often the day following the procedure, some patients are still feeling very uncomfortable. So colonoscopies, if they can be avoided, if they're unnecessary, we should be aiming to do that for the patient, not only to reduce the side effects they may experience, but also for other impacts on patients, such as they could lose 3 working days. And if you're self-employed or paid by the hour, that could have significant financial impact on you. And also there's the environmental impact of colonoscopies that we need to look at. Colonoscopies have got a significant impact on the environment. This is the waste from a single scope alone. And in the USA, they find that the endoscopy footprint per annum is equivalent to almost 86,000 metric tons of CO2. There's other impacts on the environment as well, such as the decontamination procedure. So we not only need to identify these patients that require colonoscopies to stop the low-risk the patients classified as low risk, to prevent the ones that are wrongly classified as low risk missing out in their colonoscopies, but also in terms of patients that are getting colonoscopies. These are unpleasant procedures. They have side effects. They have an impact on the environment. So there's 2 arms to this problem. What INSIZE 1, Integrated Technologies for Improved Surveillance, aims to do is to combat this. So in the UK, we're currently doing over 100,000 surveillance colonoscopies each year, and this is predicted to increase by 30% over the next decade. This is simply unsustainable. They are not only expensive and invasive, but they also are unnecessary. So INSIZE aims to improve this BSG guidelines so that we can appropriately risk stratify patients to reduce costs, but also more importantly, to improve patient outcomes. Therefore, we aim to develop a polyp risk stratification tool that improves upon the current BSG guidelines, and this will reduce the need for polyp surveillance in the low-risk patients and enable us to increase targeted surveillance for the high-risk patients. How are we going to achieve this? So we have a large team of leaders across a variety of different disciplines. So I'm the director of INSIZE and Gerard Lynch is the project manager, but none of this work would be possible without our team of talented individuals that include clinicians in the form of colorectal surgeons and colorectal pathologists, but also the clinical work that's done by our bio-repository in pulling the tissue and collecting the clinical data has been very important. We have a range of molecular diagnostics ranging from genomics, transcriptomics, IHC, and digital pathology. It's really important that we perform data integration, and this is performed by 3 teams, my own group, Philip Dunn's team, and Canon Medical Research Scotland. And we also need to look at how this new risk stratification tool would impact on the economy. So we work with a team at the University of Glasgow run by Professor Neil Hawkins to look at the health economics. And also we need to look at the end users. How do the clinical professionals that would be using this tool, how do they feel about it? What is the behaviour of patients around this, and also patient engagement? Therefore, to enable to deliver INSIZE, we have 9 work packages ranging from the administrative reporting and finance to our public and patient engagement to tissue preparation, genomics, transcriptomics, histology and IHC, digital pathology, data integration, and the actual making of the risk stratification tool itself and how that's going to sit within a clinical portal, and also our work package 9, it's health economics. So what is going to be our approach that's different from the current BSG 2020 guidelines. So the current BSG 2020 guidelines is formed on the size of the polyp, the number of the polyps, and also if dysplasia is present in the polyp. So what we aim to do is to integrate a variety of different data for the patient. So utilise all the patient's clinical data, including things such as comorbidities and age, and integrate this with their mutational landscape, their transcriptomic profile, their histology, and also immunohistochemistry markers and AI-guided computational pathology. All this data is going to be combined by the translational pathology group, Canon Medical, and the Dunn group, and integrated together to produce a risk stratification score that will sit within this risk stratification tool. It's important that this risk stratification tool is explainable. So we will have a front-facing score that the clinician and the patient can see. However, we really feel it's important that if the clinician or the patient using the score want to understand how that score has been arrived at, that there'll be various levels of explainability that they can click on using the tool to get a deeper and deeper understanding of how their score has been calculated. So that is the logistics behind what we aim to do. Where are we at the moment? So InSize 1 was launched in 2020, exactly 1 month before the pandemic. So that was great timing on my behalf. And what InSize 1 aimed to do in the first 3 years of the study was to identify the patient cohort that we could utilise in order to gather the patient information with mutational, transcriptomic and pathological data. During this 3-year period, in addition to building a cohort, we aim to get the biomarker platforms in place in terms of all the omic modalities we're going to look at, and also the histopathology in the AI. We started to develop the prototype AI method of how the data was all going to be integrated into the risk stratification tool. And we also started to gather evidence to support the clinical adoption of that. So the next few slides that I'm going to show is going to be around what we achieved in the first 3 years of InSize. So the first thing was we identified 2,642 patients from Greater Glasgow and Clyde, where we had a minimum of 6-year follow-up, so we'd be able to confidently determine whether these patients developed a metachronous polyp or not. And from these 2,642 patients, we had about half of them developed a future polyp and about half of them didn't, to give us the appropriate power to use to develop a risk tool. We also had a variety of sizes, with about 2/3 of them being over 1 centimetre in size. We had a variety of histological pathologies where only 6% were serrated and the majority were tubular or tubular villus, with about 2/3 of them having advanced features, about 2/3 being male, about 90% having low dysplasia, and we had them from all sites within the colon and the rectum, but again 2/3 were from the left side of the colon. We then took this patient cohort and using a randomised computer program, we split them into a training and test data sets, where the program was asked to equally match them for characteristics. So we had a similar number of males, females, site of disease, size, no future polyps, etc. And we had 70% of the patients, so almost 2,000 of the patients put into the training set, with around a third of the patients being put into the test set. So the training set was used to develop all of our biomarkers, and then once they were set, we validate within the test set. We also, for the polyps that were large enough, made a tissue microarray for all the patients to enable us to do biomarker screening studies, because every IHC or ISH stain we didn't want to have to use the valuable full sections. So we developed a TMA resource to allow us to identify the key biomarkers going through to the full section study, and exclude biomarkers that didn't look hopeful. For these TMAs, cores were selected by our pathologist Nuri Maka from both the basal side of the polyp and also the luminal side of the polyp, and we selected 4 basal and 4 luminal per patient. We also developed tissue classifiers. Here is an example of one of our epithelial classifiers, and here is an example of, in the yellow, the adenoma region. We then went on and developed our AI model for integrating all our data once the data was obtained. So that was our resource building exercise. We then went on to develop our omics platform. So for the first 500 patients, DNA was extracted by the molecular pathology lab in the Queen Elizabeth Hospital in Glasgow. Then this was sequenced using a gene panel of 384 genes, an adjuvant gene panel by GIA, which is now also based in the Queen Elizabeth Hospital. And then this was fed back to my own team for analysis at 1 omic level, and to Canon Medical for analysis and integration into this stratification tool. And the type of data we were getting for the patients included the type of mutations, the classification of mutations in the most commonly mutated genes. And these are the 10 most commonly mutated genes, not surprisingly, with APC and KRAS being the most commonly mutated. So we're still exploring this data in further detail to identify which gene patterns actually should be included in our risk stratification tool. But what we were interested to see, which validated our cohort nicely, was that the Wnt signaling pathway was the most mutated pathway across our samples. So as APC is in the Wnt signaling pathway, and SOX9 was also in our top 10 mutated genes, we felt we should pick a few of the most commonly mutated genes to look at in more detail. So we further investigated the role of SOX9 within our incised patients, and this was done by one of our PhD students, Sarah Albadran. So SOX9 can be full-length protein, or following a truncated mutation can be a truncated protein, which in the literature is termed as mini SOX9. And we were really interested to know that in our cohort, the majority of patients that had a SOX9 mutation all had truncated SOX9 mutations, suggesting that our patients could be expressing full-length SOX9 or truncated SOX9. And what's interesting about SOX9 is its role in the Wnt signaling pathway. So when the Wnt signaling pathway is activated, beta-catenin is released from the complex, where it can translocate to the nucleus and drive transcription. However, in the presence of full-length SOX9, SOX9 can bind to beta-catenin and prevent it from activating Wnt transcription. However, if SOX9 is truncated and is present as mini SOX9, it is no longer able to bind to beta-catenin and act as a negative regulator of the Wnt signaling pathway, allowing beta-catenin to act as a transcription factor and induce Wnt signaling. So it is interesting that the presence of SOX9 could actually have an inhibitory impact on the Wnt signaling pathway, or it could allow the Wnt signaling pathway to proceed in an unregulated fashion. So what we did was we used an antibody to SOX9, whose antibody specificity had been validated by a single band on a Western blot, followed by cell pellets stained for cell lines known to have high or low SOX9 expression. And then we stained both the training data set and the test data set using immunohistochemistry, and this was scored on the QPath platform. And what we observed was that patients that had higher levels of cytoplasmic SOX9 expression, and this was us not differentiating between full length or truncated SOX9 because the antibody was to the C terminal of the protein. But what we've seen was that not only was SOX9 associated with poor clinical outcome measures and in the binary fashion associated with metachronous polyp risk in both the training and the test set, it was also associated with patients developing metachronous polyps significantly quicker. We then combined that with the BSG to have a combined score where we had patients that had high SOX9 and high BSG being high-risk patients, and patients with low SOX9 and low BSG 20 being low-risk patients, with the remaining patients being in this intermediate subtype. And when we did this, we've seen that we could improve on the current risk stratification of the patients with BSG alone. So here we can see how BSG alone risk stratifies the patients compared to how SOX9 risk stratifies the patients. And when we combine both of these, we can see that BSG alone, high risk group is here where the adding in SOX9, it shifts this high risk group further up. So we're identifying more high risk patients. And we've seen this not only in the training set, but we've also seen a very similar trend in the test set, where the BSG high risk group is here on the red line, and by adding in SOX9, we push it further up. So what we were able to see in INSIZE 1 is by using the information from the mutational data to identify a gene to look at in more detail at protein level, by integrating only one protein with the current BSG guidelines, we're able to improve in the current BSG guidelines. So we're really hopeful that when we integrate all our data into the final risk stratification tool, that we'll be able to further improve in that. We also thought it was important for colorectal polyps to also look at proliferation in INSIZE 1. So we used Ki-67, as that's a well-established marker of proliferation and could be easily translated into the clinic. And what we've seen was that when we looked at normal crypts, we had a nice range of proliferation and index across the full polyp. But when we looked at adenomas, what was really interesting to see that the majority of the proliferating cells were at the luminal edge, with very few being seen in the basal region. And when we did heat maps of our polyps to look at this, we were able to see that Ki-67 density was much higher in the luminal region versus the basal region. So we then stained our polyps for Ki-67, and we scored them in two ways. We scored only the luminal region, and we also scored the polyp as a whole, because we're constantly thinking of the translation to the clinic, and if our pathologists are able to just score the full polyp, it's much easier than them agreeing only to score the luminal region. And when we scored the luminal region, we got an association with the future polyp status. However, this was upheld when we scored the polyp as a whole, because the expression in the luminal region is remaining dominant. We looked at this in terms of time to metachronous polyp, and again, those patients with high Chi-67, whether in the luminal or in the total polyp, are those that develop metachronous polyps significantly quicker. We then decided that we wanted to look in the TMAs to establish if by combining 3 markers we could do even better. So because of the observation we made with the Wnt signalling, we also added in ecahedron to these studies, and we looked at it at the protein level and also at the transcriptomic level, and then we combined these 3 protein IHC markers with the BSG 2020 guidelines, and we've seen by doing this that we're able to push this high-risk group even further, so we're getting much better at identifying the high-risk patients. And what we also seen that we didn't see with SOX9 alone, we're also getting much better at identifying the low-risk patients, with up to the 3-year point, only one patient that was in the low-risk category actually developing a metachronous polyp. And when we looked at this by a forest plot, we were able to move the hazard ratio that we see with the BSG 2020 guidelines from 2.3 to combining these 3 markers with our BSG guidelines to the high-risk group having a hazard ratio of 10, and even the moderate risk group having a hazard ratio of 3, which is higher than the BSG high-risk group alone. So in summary, from INCISE 1, we were able to get our patient cohort in place, we were able to establish all our omic platforms, and we were able to show by only integrating the BSG guidelines with 3 additional IHC stains that could be improved on. So we're excited to move into INCISE 2, where we're starting to integrate all our data into a risk stratification tool. So we started INCISE 2 in April of this year, and this is funding for a further 2 years. So what we aim to do in INCISE 2 is to further refine our risk stratification tool, and also establish if at the moment all the work we've been done has been in the largest, most advanced polyp, which we term as the index polyp at initial colonoscopy, but we also in INCISE 2 aim to look at all the synchronous polyps and establish if one polyp's enough, or whether we should be applying our risk tool to multiple polyps. We plan to upscale our histopathology and our omic biomarkers, and we plan to greatly expand our stakeholder engagement by having a work package totally dedicated to clinicians and patients, and we've received further funding in addition to our Innovate UK funding from Bowel Cancer UK to enable us to really unpick the opinions of the clinicians and the patients to ensure that our risk tool would be acceptable to the people that would be using it. So for our first objective of INCISE 2, we have refining the components of the risk stratification tool. So we do have gaps in our current database, and these include that our transcriptomics have been performed in bulk RNA-seq and no spatial analysis has been done. So we'd like to expand into some spatial transcriptomic study. we have only done a small range of biomarkers at the IHC level, SOX9, CD3 for pan-lymphocytes, ecahedron and Ki67, and our genomics panel has only been performed in 500 of the patients where our IHC, our transcriptomics and our clinical data is in the full cohort. So for objective 1, we want to see if we can improve our risk stratification tool by doing some spatial transcriptomics, particularly on the dysplastic regions, looking at additional IHC markers, and we hope these IHC markers will be provided to us by the mutational work and also from the transcriptomic work, because with the translation to the clinic, it's much easier to translate an IHC stain to the clinic than doing complicated omic panels. And we also aim to expand the number of patients that we do the genomic cancer plus sequencing on. So here's how it's going to look. We're going to do genomics on a further 400 index polyps. We're going to macro dissect our dysplastic regions to get some more spatial information, and then we're hoping to also go forward with some spatial platforms such as the nanostring cosmic platform, and we're going to expand the number of markers we've looked at the IHC level. And also, along with this, we're doing computational pathology on the H&Es to try and get more information from the H&Es themselves. And this additional data is all going to be integrated when we're developing our AI integration model for our risk stratification score that will sit within our risk stratification tool. Objective 2, to determine if the largest, most advanced polyp is sufficient for defining our risk. So as I've just explained, the 2,642 patients that's currently in our data set, we've used only one polyp in our studies to date. However, we have no evidence that that is the correct thing that we should be doing. So we want to, for 100 patients, identify the other polyps that were present within the colorectum at the time of initial colonoscopy, and test whether a risk stratification tool remains the same on the other polyps, or if our largest, most advanced polyp gives a higher risk or a lower risk. This will tell us if it remains the same or the index polyp gives a higher risk, that we can only apply this tool to the index polyp. However, if any of our synchronous polyps have a higher risk score using a risk stratification tool, then it's obvious that one polyp's not sufficient for analysis. We would need to do all the polyps that are excised at index colonoscopy. So where are we with developing this synchronous cohort? So we identified 100 patients where we already had all the genomic, transcriptomic and IHC markers available, and we also had more than one polyp at index colonoscopy, and we have got our clinical data in place for those 100 patients. We've identified the blocks for the synchronous polyps for those 100 patients, which has given us a further 300 blocks. So we have around 400 polyps in all to study for these 100 patients. We have started cutting the sections for this cohort, and we have extracted the DNA, and we're halfway through sequencing these 300 additional polyps. The areas have all been annotated and are ready to go to Bioclavis for the 10% transcriptomic analysis, and we've started performing the IHC and applying our H&E markers. So for INCISE 2, we're hoping to be at a stage further on. In addition to knowing that we can improve in the BSG 2020 guidelines, we will be in a place where all our data will have been integrated, and we will have our final parameters defined that are going to be in our risk stratification tool, and we will have that optimized. We will also have a prototype risk stratification tool in place, which Canon Medical have already started engaging with our PPI group to get their feedback on what our tool looks like. So that will be in place, and we'll also have the PPI work package with the healthcare professionals and the health economic studies established to actually demonstrate the feasibility and the impact of our risk stratification tool. By the end of INSIZE 2, we're hoping to be in a place to apply for follow-on funding to perform INSIZE 3. What INSIZE 3 is going to look like is we are going to do 3 more retrospective studies across 3 independent health boards in 3 geographically distinct locations, 1 hopefully in Northern Ireland, 1 in England, and 1 in Europe, and we've already started those discussions with collaborators. And alongside that, we're going to perform a proof of concept study within Glasgow, Greater Glasgow and Clyde, which will be a small prospective study to actually demonstrate that in the real world, the data required to inform our risk stratification tool can be easily identified and combined in a tight, timely manner. So I would like to finish there by thanking all members of the INSIZE collaborative, in particular our key partners. So there's my own group, the Translational Cancer Pathology Lab, but there's also the Greater Glasgow and Clyde Bio-repository, Oracle Bio, that is our digital pathology partners, BioClavis, that's our transcriptomic partners, and Canon Medical Research that have been leading on the AI-based data integration and also the making of the actual tool itself. And we've got some pictures here of us with the partners at various different events, and here's the INSIZE group that sits within my own lab. Thank you.
Thank you very much indeed, Joanne. That was a really fantastic talk. I enjoyed that tremendously. So thank you very much. If we were doing this live, we'd be hearing the sound of lots of clapping now, but we'll just have to imagine that. Okay, so it's time for the question and answer session. I'm going to start off, if I may, and I'd like to start off with a somewhat philosophical question. Can I invite you to say something about why you think a current polyp that a patient has now is going to tell you something about the risk of a polyp that hasn't yet formed but will be formed at some point in the future in a different place in the bowel?
Okay, so it's based around the sort of field change effect that we feel that an individual patient not only has a prior risk from the sort of work that Malcolm Dunlop's been doing with his risk prediction tools for cancer itself, but also there is a field change across the full bowel that makes these patients more likely to develop a polyp in the first place. And that combined with the actual data, the clinical data for the patient, like their lifestyle choices, their BMI, their comorbidities, these all combine together, and it might not be that that particular polyp has developed in that part of the bowel because it's that one tiny part of the bowel that's got a predisposition to the polyp developing. It needs to be, we look at the host in general. Even things like systemic inflammation could be impacting on whether these patients are likely to develop future polyps. So it's important for us to get all the information we can from that particular polyp, but it needs to be integrated with the data on the patient as a whole.
Okay, lovely. That's a good answer. Thank you. So I'm now going to turn to the Q&A box and read out some of the questions that audience members have asked. So starting with Nick West, who says, sorry if I missed it, but in addition to stratification of risk in adenomas, are you also looking at risk specifically in serrated lesions with and without dysplasia?
Yes, so we've only got 6% of the patients in our cohort are serrated lesions with only about 100, so that's only about 140. So we're looking at them quite carefully. So from the patients, the 140 patients that were initially identified from the pathology reports as of having serrated lesions, Nurimaca has recently went back and reassessed all those H&Es, and if you're looking for a true SSL lesion, we only have around 50 of them at the moment. So we're very, very interested in these lesions, especially with dysplasia or without dysplasia, and we're doing much more intensive investigations. These, in fact, we've just got a recent CRC project grant with Seth Cofelt to look at the immune landscape, in particular the gamma delta T cells for this lesion type. But we don't have the numbers, but we are very interested in them and we're looking at them in more detail.
Okay, thank you. So our next question is from Sarah Aitken, who says, thanks for your talk, Joanne. It looks like this will create a very rich data set. I was interested that 2/3 of your BCSB polyps are greater than 1 centimetre in size. This is much larger than we see. Does your risk stratification tool with SOX9, MIB1 and cadherin perform equally well in different size polyps? And a related question, how did you pick which one polyp to include for each patient?
Yeah, so we're worried that our cohort is heavily biased towards larger polyps. So one of the most important studies we do when we do our Kaplan-Meier risk curves, we also do Cox regression, and we look to see if it's independent. And the main parameters we're looking at in that is, does it work in small and large polyps? Does it work in patients with only one polyp compared to those with multiple polyps? Does it hold up in men as well as females? And does it hold up across the different histological subtypes? So we are aware that the cohort is biased towards the larger polyps and also towards left-sided polyps. So these are the considerations that we look at when we're making sure that the markers we include should be included. And how we actually identified the first index polyp was crude. It was the largest, most advanced polyp at index colonoscopy. That's why it's really important to go back and do this follow-on study looking at synchronous polyps, because that might not be the most appropriate polyp to use.
Yep, thank you. Okay, so our next question is from Graham Murray, who says, many thanks for an excellent talk. Is there potential for blood-based biomarkers to contribute to risk stratification?
I think very much so, and that would be really great because it would get around all the problems associated with colonoscopies. We've not built in a blood biomarker arm to the INCISE study, but I think it's definitely something that should be looked at.
Might that be part of a future study, INCISE 3 or 4 or some other study?
Yeah, I think we need to look at it. I don't think shed DNA or circulating DNA will be the answer, because I think it needs to be more advanced tumours for that. But I think there will be markers. One of the ones that I think might be the lowest hanging fruit is looking at something like CRP for systemic inflammation.
Okay. Okay, thank you. So Nick West comes back with a second question. Are you looking at the microbiome in addition to tumour markers?
So we have performed a study where we've done RNA scope for 16S and for fusobacteria, and we have also sent a subset of our polyp to Iris Naglital's lab to do her biofilm biofilm assay on for the microbiome, and Sarah, the PhD student, has recently got a small endowment grant from GGC Greater Glasgow and Clyde to do some spatial transcriptomics. So in the areas where we see the fusobacteria, is the transcript of the surrounding cells different in the polyp tissue from the polyps that have no fusobacteria or are distant from the fusobacteria. We have been quite narrow in that we've concentrated in fusobacteria, and we're very well aware that there is a whole host of bacteria that we haven't looked at, but we've chosen, because of Susan Bullman's work, to start with fusobacteria.
Okay, thank you. The next question is from Ian Tomlinson, who says, you may have said this, sorry, I had some connection problems, but SOX9 is a driver mutation in colorectal cancer and quite possibly in polyps. How do the mutant samples fit into the expression models?
So I think what he's asking is, do the mutant samples result in an increase in protein expression? Is that?
I think that's part of the question, yes.
Yeah. So at the moment, that's what we're doing. So we've just had so we had the information for quite some time that SOX9 was one of the most commonly mutated genes in the cohort, and that's what instigated us doing the IHC experiments. But we've just got the data back on what all these different mutations are and the different types of mutations. So we're looking to see if the patients that had the truncating mutations are those the ones that are overexpressing IHC, etc. And we're also looking to further understand the driving role of SOX9 in the adenomas and the colorectal cancer models. We're working with Owen Sampson, who's working on mouse models at the moment with us, and Sabine Tetcher's also really interested in SOX9. So at the mechanistic level, we're exploring that further at the model level with Owen, who knows much more about these things than me.
Sure. So Ian has come back to clarify and says, are the SOX9 mutations predictive?
So if we combine in the subcohort of patients that we've got SOX9 mutations and ARID1 mutations, they are associated with future polyps. But when we look at SOX9 alone at the moment, there's only a trend towards it, but we've only got the SOX9 data in 500 patients at the moment, but we'll soon to have it in 900. So then we'll be able to answer that question with more certainty.
Okay, thank you. So Annie Baker asks, do you have any insight into whether the immune environment is different between high risk and low risk polyps, either from your IHC or the whole slide transcriptomics?
So at the moment, we've done CD3 in the full polyps, but we've also had a look at CD4 and FOXP3 in a subset of polyps, and there is definitely a different immune landscape in the ones that have the higher risk. And Stephen McSorley, who is the clinical lead in NSAIS, is currently developing his fellowship proposal to look in more depth across the immune landscape. When he looks at the CD3 high and lows in terms of transcriptomic profiles, we're seeing nice differential gene expression as well. So this is, but we've not really touched on any innate work at the moment, so that this is, Steve wants to really look at this in depth for his fellowship proposal that he's hoping to submit within the next year.
Yeah, I'm sure that will be a very interesting study for him. Okay, so another question from Nick West, who says, really interesting talk, thanks. If the current BSG risk tools are imperfect, specifically the failure to identify high-risk patients with small lesions, how comfortable are you with the new resect and discard policy of the bowel cancer screening program?
So not very comfortable.
Yeah, I would agree.
Yes. Yeah. Not comfortable is the quick answer to that. I think it's the wrong approach, but we need more evidence to demonstrate that to them.
Yeah, I think we're at risk of throwing away very important information, and you're absolutely right. I would agree. We need the evidence to prove that case, and make a change there. Okay, so here's a question from a colleague of yours, Philip Dunn, who says, thanks, Joe. Some of the data you presented for risk stratification biomarkers are biology-based, as in genes, mutations, etc. While I love a biology-based biomarker, would an unexplainable AI tool that performs well be more acceptable to the pathology gastro field, in your opinion, than an explainable yet lower predictive value of a biology-based explainable tool?
Obviously we want the best tool we can get, but I'm really uncomfortable with black box AI methodology. I would rather have a tool that performs slightly less well, that we totally understand how it performs, because if it's a black box based AI tool, how can we discuss it with the patients and explain what it means? And we can't even give patients any advice. Patients want to know, can I change something, for example? And if it's a totally unexplainable tool, you can never do that. So in my opinion, I would much rather have an explainable yet lower predictive value biology tool. And by understanding how that risk comes to, we can also open up the avenue for ways of intervening, even if that could be some sort of prophylactic therapy, if there's pathways with drugs that are very low in toxicity, etc.
Okay, that's a great answer, thank you. So it's very nearly 10 o'clock and we have a tradition of finishing on time so people can have the rest of their morning back. So I'm going to end the question and answer session there, and I'm going to finish off by thanking you once again for a brilliant lecture. I really enjoyed it, and also quite a lively question and answer session at the end as well. So thanks very much indeed, Joanne. Thank you.
Thank you. Bye.
That's all. Thanks everybody for attending. Thank you. Bye.